Hi, Laura Roselli here. Today we're going to talk about COVID tests, and we're going to talk about three tests altogether. Two have to do with whether you're currently infected with coronavirus 2, and the third is whether you were in the past exposed to the coronavirus. The first test we're going to talk about is called a rapid test or an antigen test. This is a test where you'll get the results in about 15 minutes. And what we're looking for is to see whether you specifically have the virus in your system, and you'll do a nasal swab. That's where the virus will be living. And what we're looking for in the rapid test is specifically these proteins here. These proteins on the outside of the virus are called spike proteins, and they are referred to as an antigen because they make your immune system respond in some way. So what we do is we do the nasal swab. We then take that sample from the nose and we put it on a cartridge about that big. And then there will be some interactions between the potential antigen and some other antibodies that are sitting on the test strip. If there is a positive interaction between the two, you're going to see a line that comes up on this strip, much like a pregnancy test. And that would indicate that you're currently infected with SARS coronavirus 2. Now, the other type of test is called a molecular test or PCR test. This one takes longer. It has an advantage of being a little more accurate, and it can pick up an infection earlier in the exposure time frame for the patient. In this test, we have the virus as well, but instead of actually trying to identify these spike proteins, we're going to do a nasal swab, send it off to a lab, and the lab technicians are going to crack this virus open, and they're looking for the genetic material that's inside. This is RNA. RNA is very similar to DNA that we have in our chromosomes that make up our genes, but the virus just uses a different language for their genes. So what we want to do is the technician is going to remove this RNA, and then we're going to try to figure out whether this RNA is present in your nasal swab or not. And in order to determine it, you cannot actually just use one piece from one virus or two viruses. It's just not enough. So we're going to do something called PCR. In PCR, I'm going to take this virus, I'm going to put it in a test tube or a tube that's much smaller than this, but for illustrative purposes, the RNA of the virus gets put into this tube. We add some other reagents, and we go through a process called PCR. And PCR just makes copies. It takes the first copy and makes 2. Then from there makes 4 and 8 and on until after a couple of hours, there are many pieces of the RNA, copied pieces of RNA from the virus that was in your nasal swab. And this is enough RNA to be detected in a test. This is the molecular or PCR test, and this is more accurate because, as we said, it can pick up earlier in the life cycle of the virus within a patient. It can pick up the presence. The third test is an antibody test, and an antibody test tells me whether my body has ever seen SARS coronavirus 2 before. And this is taken with blood, and what they'll do is they'll take a look at your blood and see if you actually have antibodies floating around in your blood that actually match with these antigens, the spike proteins. That doesn't necessarily mean that right now you're infectious or you're sick. What it means is that at one time your immune system has seen this virus and you fought it off, and you have immunity to it in the form of these antibodies. So there are the three tests: the rapid test, the PCR test, and then post-infection and antibody tests will tell you if your body has seen this virus before in the past. Hope this helps. The next time we're going to talk about the different types of vaccines for COVID-19. Take care.